So what happens linking carbohydrates to amino acid? So what happens if your body requires alanine, a monomer or an amino acid? So amino acid are breakdown of proteins. Amino acid are like monomers. When they unite, they form polypeptide bonds or peptide bonds that will now form what? Proteins. So when your body requires an alanine or non-essential proteins, it will now undergo another process, and it is stimulated by an enzyme. In the process by which you form an alanine from pyruvate, it is stimulated by an enzyme known as alanine transaminase. Alanine transaminase. What's it doing? It's converting pyruvate to what? Alanine. By taking what? By adding pyruvate and glutamate. So glutamate and pyruvate we add together to form alanine and alpha ketoglutarate. This alanine is depending on what the body needs for that moment, and this alpha ketoglutarate can go into the Krebs cycle and continue the production of NADH and FADH that will enter the mitochondria to produce energy. So the equation you have to take home is pyruvate reacts with glutamate to form what? Alanine plus alpha ketoglutarate in the presence of what? An enzyme known as alanine transaminase. So this is a key reaction in glucose aniline cycle, which transports amino group from muscle to the liver during a fasting state. Alanine transaminase is a key liver function test maker. So replenishing the tricarboxylic acid cycle or the Krebs cycle. What happens? So when your body needs oxaloacetate, and oxaloacetate is an intermediate in the tricarboxylic acid cycle, so what your body will do is to start converting this pyruvate to oxaloacetate by an enzyme known as pyruvate carboxylase. Don't worry, you understand this better in tricarboxylic acid cycle. But for now, we are still on pyruvate metabolism. So just keep on watching and keep on following the playlist. You understand this better as you proceed forward. So pyruvate reacts, or pyruvate is converted towards oxaloacetate by an enzyme known as pyruvate carboxylase. We are talking about pyruvate metabolism, which pyruvate will undergo 4 different processes depending on what your body needs. If your body needs ATP, it will undergo acetyl in aerobic respiration. If your body does not need energy, or if your body needs energy but there is no oxygen, it will now undergo the formation of lactate by lactate dehydrogenase enzyme stimulate that formation, and that process is anaerobic. If your body needs aniline, your body will now convert this pyruvate to aniline by an enzyme known as aniline transaminase. Now, if your body also requires oxaloacetate, which will now enter into the Krebs cycle, your body starts converting this pyruvate to oxaloacetate. And during this process, an enzyme known as pyruvate carboxylase converts pyruvate to oxaloacetate. And in pyruvate carboxylase, we are taking ATP molecule and we are forming ADP plus phosphate. And one thing you should also know is, in pyruvate, remember, pyruvate is a 3-carbon molecule, and pyruvate is a product of glycolysis by which glucose is converted to pyruvate, which is pyruvate is a 3-carbon atom. So one glucose held 2 molecules of pyruvate, which is 3-carbon atom. Now, pyruvate also is stimulated by a vitamin known as biotin. So this is the vitamin biotin, and this pyruvate is converted to oxaloacetate. And recall that pyruvate is a 3-carbon atom, and this 3-carbon atom and oxaloacetate is a 4-carbon molecule, and pyruvate is a 3-carbon molecule. Molecules are like monomers of atom. Molecules are gotten from when you combine atoms together, and atom takes part in a chemical reaction, and it can be created and it can be destroyed in nuclear energy. The energy of an atom can be converted from one form to another. Don't forget that is our basic chemistry. So 3 carbon atom is 4 carbon atom. These are molecules. So an enzyme known as pyruvate carboxylate converts pyruvate to what? To oxaloacetate, which is 4 carbon atom, by what? Adding carbon in it in form of carbon dioxide. Adding carbon in it in form of carbon dioxide, and this is called what? Carboxylation. So carboxylation is a process by which we add carbon in form of carbon dioxide in a molecule or in a substrate. Now we are carboxylating pyruvate, which is 3 carbon atom, to now form oxaloacetate, which is 4 carbon atom, which is now going to the Krebs cycle to produce energy or undergo so many processes depending on what your body needs. So the equation you have to take home is pyruvate reacts with carbon dioxide plus ATP molecule to form oxaloacetate, ADP, and phosphates. So that is what you have to have in mind. Once you have the equation, you can further go ahead and draw the molecules. So this is a primary anaplerotic reaction. It replenishes oxaloacetate and other tricarboxylic acid cycle intermediates that are consumed in the biosynthetic pathway. Regulation: allosterically activated by high level of acetyl-CoA. So this process is activated by acetyl-CoA. So when we have too much acetyl-CoA, our body will not like to keep on converting this pyruvate to acetyl-CoA in the presence of oxygen or in aerobic respiration. So our body will now devise a means of converting this pyruvate to oxaloacetate so that we can store this fuel, so that we can store these nutrients. And this process is allosteric. Look at what I mean. Remember, if you have done enzyme or enzymatic reaction, enzyme has a structure that looks like this. And in allosteric regulation, the acetyl-CoA, so this is the active site of the enzyme. So the active site is where the substrate bind, but acetyl-CoA will now come allosterically. So when we talk about allosterically, it means when a substrate is not binding to the active site of the enzyme, it is binding at the allosteric site. So this is the allosteric site, this is the site. So the acetyl-CoA will come and bind to this allosteric site. So this allosteric site is what we call the allosteric activated by high level of what? Acetyl-CoA. So when substrate is binded to an enzyme to perform a specific reaction, that is called allosteric reaction. We have the summary table